In the interest of time, I think we are going to move on to the next speaker, who is Dr. Erinn Rankin, an Associate Professor of Radiation Oncology and Obstetrics and Gynecologic Oncology.
Sorry about that. So thank you very much. I'd like to start by thanking Dr. Oliver Dorigo for organizing this important symposium, bringing together our gynecologic oncology community, both here at Stanford and around the world through our online participants, really in honor of Dr. Barrick. And so thank you very much for this opportunity to really share with you the research in our lab where we're understanding how we can exploit the unique ovarian cancer tumor microenvironment for cancer therapy. As we've already heard today, ovarian cancer remains a leading cause of cancer-related deaths among women in the United States as well as worldwide. And it's a particularly deadly cancer because the majority of patients are diagnosed at advanced distant metastatic disease. And at this stage, when patients are diagnosed with distant metastatic disease, they have very poor survival rates of about 30% due to treatment resistance and recurrence. And so there's a really urgent unmet clinical need to identify and develop more effective therapies for the treatment of ovarian cancer. And this is an area that we're really focused in on. And so as a cancer biologist, I'm really interested in understanding this unique peritoneal metastatic microenvironment that ovarian cancers grow and become resistant to therapy in. As we've already learned and heard about today from Dr. Howitt, ovarian cancers arise in the fallopian tube and then end up spreading to the ovary and then gaining access to the peritoneal fluid, circulating around the peritoneal cavity, where eventually the cancer cells and spheroids will attach to many different tissues and organs in the abdomen and begin to grow and form metastatic distant lesions at these sites. And interestingly, at these sites, all of these peritoneal surfaces and tissues are lined by a unique layer of cells called mesothelial cells, shown on top here. You can see these are mesothelial cells lining a human omentum, and they're often the first point of contact when ovarian cancer cells circulate and form distant metastases at these sites. You can see these cancer cells in an early stage at the human omentum interacting with these mesothelial cells. Interestingly, in addition to lining all of the peritoneal surfaces in the abdomen, mesothelial cells also make up about 10 to 80% of the cellular content in patient ascites fluid. So they're an important stromal component of the ovarian cancer tumor microenvironment. And we and others have contributed to work where we've shown that these mesothelial cells can contribute to the early stages of ovarian cancer metastasis in terms of facilitating adhesion, proliferation, migration, invasion at these distant tissue sites. However, the role of mesothelial cells in the later stages of disease, particularly in chemoresistance, which is a major clinical problem in ovarian cancer management, really was not known. And so a postdoc in my lab, Jin Kwan, set out to investigate the question whether mesothelial cells contribute to ovarian cancer resistance, and if so, how, and could we therapeutically intervene there? So to ask this question, what he did was first just tested and investigated whether the presence of cancer-associated mesothelial cells, shown by CAM here or LP9, would facilitate ovarian cancer resistance in animal models. So we injected ovarian cancer cells alone or in combination with patient-derived cancer-associated mesothelial cells, and then treated them with cisplatin or control. And you can see that cancer cells alone responded to cisplatin treatment. However, quite shockingly, the ovarian cancer cells that had been co-injected with the cancer-associated mesothelial cells no longer responded to cisplatin treatment. And we've seen this with multiple different cancer cell lines and patient-derived cancer-associated mesothelial cells. And you can see this is again reflected here in the primary tumor endpoint weights as well, demonstrating and identifying an important role for cancer-associated mesothelial cells in ovarian cancer resistance. We then wanted to determine if this was indeed mediated through secreted factors. And so we could use in vitro culture systems where we can culture cancer-associated mesothelial cells on the top and cancer cells on the bottom, culture them together for a period of time, and then expose the cancer cells to different platinum-based chemotherapies. And we saw that indeed secreted factors produced uniquely by cancer-associated mesothelial cells were facilitating platinum-based chemotherapy resistance. So you can see compared to the control treated cancer cells that respond to different doses of increasing doses of cisplatin, those that were treated with cancer-associated mesothelial cells have blunted responses to these chemotherapies. Interestingly, this property is unique to mesothelial cells isolated from cancer patients. Because when we compared this to mesothelial cells isolated from patients with benign non-cancerous disease, they did not have the ability to promote chemoresistance. So cancer mesothelial cells are associated mesothelial cells are secreting factors that are facilitating a chemoresistant phenotype in ovarian cancer cells. And so we became really interested in exploring what those secreted factors are and can we potentially therapeutically target those to help overcome chemoresistance in ovarian cancer. So we set out by profiling the secreted factors through array approaches from benign mesothelial cells or cancer mesothelial cells to identify those factors that are enriched in the cancer-associated mesothelial cells. And one interesting factor that was increased in red here in the cancer-associated mesothelial cells was osteopontin, or OPN. We found and validated that indeed osteopontin was increased both at the RNA and secreted protein levels in these cancer-associated mesothelial cells. We also looked at independent gene expression profiling data sets, and again found this was a top hit of genes that are highly enriched in cancer-associated mesothelial cells compared to mesothelial cells from benign disease. We were also fortunate to have a very nice collaboration with Oliver Dorigo, and we're able to look in patient ascites samples at osteopontin expression. And we found it within the ascites, indeed, you can see there's a high number of calretinin positive mesothelial cells in this patient ascites. And indeed, most of the osteopontin positive cells in red here were actually mesothelial cells. So we found that cancer-associated mesothelial cells are expressing very high levels of osteopontin uniquely. And we found this very interesting because in the literature, osteopontin had already been described as being overexpressed and associated with poor survival in ovarian cancer. And we validated that with our own independent analyses here. In addition, we also knew that osteopontin is known to be overexpressed and play pro-tumorigenic roles in a number of other solid tumors shown here. And it promotes many pro-tumorigenic activities, including metastasis, angiogenesis. More recently, there's been a lot of interest in its role in immunosuppression and macrophages. However, its role in chemoresistance and its functional role in ovarian cancer remains largely unknown. We therefore investigated whether osteopontin was both necessary and sufficient for ovarian cancer chemoresistance. So in these studies here, we knocked down osteopontin expression from the mesothelial cells and showed that that could now restore and enhance ovarian cancer cell chemosensitivity. And in contrast, we were able to treat ovarian cancer cells with recombinant human osteopontin. And indeed, recombinant osteopontin is able to facilitate it and promote ovarian cancer chemoresistance. So we're very excited about the potential of targeting osteopontin in ovarian cancer to enhance chemotherapy responses. While there are no effective clinical inhibitors developed at this point targeting osteopontin, we took an RNA aptamer approach in our preclinical studies to test this proof of concept. So we utilized an ovarian cancer mouse model of metastatic disease. This is a syngeneic ID8 model where we treated the mice alone with vehicle, with cisplatin alone, the osteopontin aptamer alone, or the combination of cisplatin and the osteopontin targeting agent. And we were able to see that targeting osteopontin was now able to really significantly enhance chemotherapy response in this aggressive preclinical model of ovarian cancer. So we're currently very excited about teasing apart not only the molecular mechanisms by which Osteopontin is facilitating ovarian cancer chemo resistance and how this is mediated through mechanisms supported by the tumor microenvironment. And in addition, with the goal of translating these findings into the clinic, we're now independently in the lab working on developing effective antibody approaches to target osteopontin for the treatment of ovarian cancer. And this is something we're actively developing now. With that, I know I'm almost out of time, so I'm going to quickly highlight another very exciting project in the lab with translational potential that was led by two very talented gynecologic oncology fellows, Karen Levy and Stephanie Chow. And in this project, we had a really very exciting collaboration with Dr. Bill Liu's lab in radiation oncology at Stanford, where we're investigating the efficacy and safety of utilizing an emerging radiation treatment modality called flash irradiation and its ability to enhance the therapeutic of abdominal pelvic radiation therapy to enhance anti-tumor immune responses in ovarian cancer. And so while radiation therapy isn't widely utilized in the clinical management of ovarian cancer anymore, really largely due to the toxicities associated with wide field treatment of abdominal ovarian cancer metastasis, there is a lot of interest in the field in reconsidering the use of radiation therapy as a strategy to help induce anti-tumor immune responses within the tumor microenvironment. This is based off of other clinical results where we've seen that radiation therapy can promote an anti-tumor permissive tumor microenvironment and synergizes very nicely with immunotherapies. And so it's been hypothesized that perhaps radiation therapy could potentially be utilized as a strategy to help convert our cold ovarian cancer tumors into hot immune infiltrated tumors where we can promote anti-tumor immune responses through its role enhancing tumor antigen exposure as well as recruiting activated T cells within the tumor microenvironment. However, we're still left with the same concerns of the normal tissue toxicities associated with irradiating our highly radiosensitive intestine within this type of radiation field. And so we became very interested in looking at the possibility of using this emerging radiation treatment modality called flash irradiation as a way to enhance the therapeutic index of abdominal pelvic radiation therapy for the treatment of ovarian cancer. And so just briefly, flash irradiation is essentially an ultra high dose rate irradiation or delivery of radiation therapy, where conventional radiation therapy is delivered at doses at about 3 to 4 gray per minute. Flash radiation therapy is delivered at about 1,000 times faster at greater than 40 gray per second. Actually, most of our work is at 200 gray per second. And over the past decade, this now emerging radiation treatment modality has really been shown to be highly effective in enhancing the therapeutic index of radiation therapy through reducing normal tissue toxicities associated with radiation treatment. So in the field, we've seen that flash radiation can maintain tumor control in multiple different tumor models while reducing associated normal tissue toxicities. And this is now being seen across multiple institutions around the world in multiple different tissue sites, as well as in multiple species. So it's been seen in mice, companion animals, and human clinical trials are now ongoing. Our work in collaboration with Bill Liu's lab here at Stanford, who's a pioneer in this flash irradiation space, and that he developed one of the very first flash irradiation preclinical treatment systems. We really worked together to demonstrate the efficacy and safety of flash irradiation in these preclinical models and looking at intestinal injury. And just to summarize here, because I know I don't have the time to take you through all the data today, but what we've been able to observe and we're really excited about is flash radiation can reduce radiation-induced intestinal injury in both animal models, and we've worked with Sarah Halsoer in looking in intestinal human enteroid models where we see enhanced regeneration as well. Flash irradiation therapy, in contrast, though, is able to maintain metastatic ovarian cancer tumor control in very aggressive preclinical models of ovarian cancer. And most importantly, we were able to demonstrate, we were the very first group to demonstrate that flash irradiation can safely combine with immune checkpoint inhibitors, such as anti-PD-1, as a combination treatment modality. And we demonstrated this in ovarian cancer, where it can enhance infiltrating activated CD8 T cells into the ovarian cancer tumor microenvironment. So we're also very excited about the translational potential of this work. And we have two arms where we're looking more into the mechanisms by which flash irradiation protects and reduces toxicity in normal tissues, such as the intestine, and how it's actually facilitating the recruitment and activation of T cells in the tumor microenvironment. And in parallel, we're collaborating with Bill Liu and his team, where they're developing clinical flash irradiation treatment systems called phaser that we hope to one day work towards translating for the treatment of different gynecologic cancers. So with that, I'd really like to thank and acknowledge all the members of my lab who've contributed to this work. I tried to highlight the folks involved directly in the projects I talked about today during the presentation. And I'm very grateful for the collaboration and support of our colleagues, including Oliver Dorigo in gynecologic oncology, Bill Liu in radiation oncology, Sarah Halsorn in bioengineering. And finally, I'd like to really thank and acknowledge Dr. Barrick for all of his mentorship and support, really for your warm welcome into the ovarian cancer research community here at Stanford, and really for all of your support and mentorship, and has been so instrumental in helping my ovarian cancer research program grow and be successful. And I'm so grateful and thankful. Thank you.